With the rapidly expanding field of nanotechnology, it is important to understand the possible toxic effects which nanoparticles can have on living organisms. Gold nanoparticles are submicroscale gold structures which are found in nature and are synthetically made. Gold nanoparticles are capable of doing a lot of functions which normal gold in bulk cannot do. It is often difficult to grasp how small a nanoparticle really is. The width of a single human hair is about the same size as 500 gold nanoparticles. Nanoparticles can be engineered or found in nature. Engineered nanoparticles can come in a variety of shapes and sizes, along with various chemical properties. Gold nanoparticles can be found naturally in weathering gold deposits. Gold nanoparticles can be used in the electronic industry, the drug delivery systems, molecular diagnostics, and cancer treatment, among other things. However, their toxic effect is still questionable. Gold nanoparticles can be taken up by organisms intravenously, intramuscularly, orally, intrarectally, and through inhalation. Thankfully, in this study, we are only investigating the inhalation. Inhalation is most likely to occur when experimenting with or creating the nanoparticles. Inhaled gold nanoparticles do not generally make it into systemic circulation. This is dependent on their size and other characteristics which we will investigate. Once inhaled, gold nanoparticles attach to the respiratory membrane of the alveolar epithelium. The nanoparticles rapidly interact with surfactant protein D, causing slight aggregation of the gold nanoparticles. Gold nanoparticles then interact with a receptor, which induces endocytosis of the aggregated nanoparticles. The exact receptor varies depending on the surface coating of the nanoparticle, which is discussed later. Receptor-mediated endocytosis is largely affected by physiochemical properties of the golden nanoparticles. The properties are the size, shape, and coating of the nanoparticle, while cellular temperature also seems to have an effect on the endocytosis rate. The small size of gold nanoparticles allow them access to most cells and even subcellular components. Gold nanoparticles can be made in a range of diameters from about 2 to 50 nanometers. When less than 5 nanometers, they are able to easily translocate through diffusion from lung tissue into systemic circulation following inhalation. However, larger nanoparticles are kept in the lungs and less than 1% enter circulation. Endocytosis can be inhibited by using sharp-shaped gold nanoparticles. Spherical gold nanoparticles, however, are the best absorbed. Two of the most common seen gold nanoparticle shapes are the nanorod and the nanosphere. Gold nanoparticles can be engineered with specific functional groups which affect the properties of the nanoparticle at the surface. PEG is commonly used to mask the immune recognition of gold nanoparticles. Other ones could be carboxylic acids, amines, thiols, just about anything you want. Overall, the absorption by physiochemical properties can be determined using the size of the nanoparticle, the shape of the nanoparticle, the surface coating on the nanoparticle, which in close relation is to the charge of the nanoparticle. If the nanoparticle is loaded with amines, for example, at certain pH levels you could have a protonated positive cationic charge. Studies have shown that at cold temperatures, endocytosis is strongly discouraged of gold nanoparticles. This is likely due to the energy dependence of kinetic motion. Unless the golden nanoparticles are less than 5 nanometers in diameter, they are kept in the lung surface tissue and are picked up by the alveolar macrophage. They do not translocate into the bloodstream, and therefore they are trapped inside and do not reach systematic circulation. Since the gold nanoparticles are kept trapped in the lung tissue, there is no systemic circulation, and therefore there is no reaching of the gold nanoparticles to the liver through inhalation. This means there is no relevant first-pass metabolism of the gold nanoparticle. When gold nanoparticles are taken up by the alveolar macrophage, the macrophage attempts to break them down as a foreign particle. The alveolar macrophage then engages in a function known commonly as the respiratory burst. This process begins directly following endocytosis and involves the breakdown of glucose into the NADPH molecule. Following the endocytosis of gold nanoparticles by the alveolar macrophage, which is also called phagocytosis, glucose is metabolized down the pentose phosphate pathway, producing multiple molecules of NADPH. Cytochrome B from the granulocyte-specific granule combine with NADPH oxidase, activating the protein. To be more specific, cytochrome B, shown in the red, is activated by P47, P67, P40, and RAC2 to activate an NADPH oxidase. Activated NADPH oxidase enzymes use molecular oxygen to oxidize the NADPH formed in the metabolism of glucose. This oxidation produces superoxide anions in the alveolar macrophage. This is called oxygen-dependent myeloperoxidase-independent intracellular killing, and this portion of the diagram is not included. Superoxide anions are then converted to hydrogen peroxide and oxygen by superoxide dismutase. Superoxide anions can also react with hydrogen peroxide, forming highly toxic hydroxyl radicals and single oxygen. This causes oxidative stress in the alveolar macrophage. Reactive oxygen species inside the alveolar macrophage inhibit IKK. This allows nuclear factor kappa light chain enhancers, NFKBs, to encourage the transcription of DNA in the cells. This encourages the cell cycle progression. Reactive oxygen species can also activate NFKBs to increase the levels of pro-inflammatory cytokines such as IL-8, TWF-alpha, or MMP9 to activate histone acetyltransferases. This will open up the chromatin structure of DNA and allows for the inflammatory genes to be transcribed. The method of this is not completely understood and is still under investigation. Reactive oxygen species can also inhibit PTP from closing the growth factor channel by phosphorylation. This causes downstream effects which lead to the activation of growth factors by ERK, extracellular signal regulating kinases. This not only encourages cell growth, but also inhibits SMAD. Inhibition of SMAD inhibits cell cycle arrest and apoptosis. Inflammation in the airways can cause difficulties breathing, similar to the effects of asthma or chronic obstructive pulmonary diseases. Progression of airway blockage can even lead to be bad enough to cause disability and death. However, with the limited testing in vivo of gold nanoparticles, it is unknown if the severity of inflammation could ever cause such effects. In addition to the inflammatory consequences of the reactive oxygen species produced by inhaling gold nanoparticles, remember that the radicals are also capable of direct damage and cell toxicity by DNA damage, lipid peroxidation, protein damage, and you know how it goes. Oxidative stress produced by the respiratory burst of alveolar macrophage causes inflammation of the lung tissue, potential inhibition of apoptosis, and the encouragement of the cell cycle. The disruption of the cell cycle does sound as though gold nanoparticles could be carcinogenic when inhaled, however research has shown no implications of this. Although gold nanoparticles may not seem carcinogenic, we can still put some pieces together. Encouraged cell growth means you have more cell division and more cell numbers. Inhibition of the cell cycle arrest, plus the ability of not apoptosing, means you probably have more cells that aren't dying. More cells in the lung tissue means you have less space in the lung tissue. Less space in the lung tissue means you have more constriction in the lung tissue. More constriction in the lung tissue obviously means you will have difficulty breathing. So, have gold nanoparticles really caused much damage themselves? Gold nanoparticles were absorbed. After being absorbed, reactive oxygen species were created in natural defense as they would be as any other toxin affecting the lungs. The reactive oxygen species have produced the discussed toxic effects, and so they are considered the effector toxin of the study. They have caused damage all over the lung tissue without any specific target. As previously mentioned, gold nanoparticles are not really metabolized. They are largely trapped by vesicles in the alveolar macrophages and other lung tissue. The exact excretion method is debatable, however they seem to be found in the feces. Trapped gold nanoparticles move through the respiratory tract by mucus developed by goblet cells. The ciliary motion of ciliated respiratory epithelial cells move the mucus towards the larynx, where it is eventually swallowed and ingested. Research investigated has not specified what happens specifically, however gold nanoparticles are known to appear in the feces. Roughly 30% of dosed nanoparticles are excreted this way every 24 hours. Reactive oxygen species generated by the inhalation of gold nanoparticles are neutralized by glutathione and antioxidant enzymes. Glutathione can neutralize reactive oxygen species by antioxidant activity before the radicals are formed. Reduction of the hydrogen peroxide precursor to hydroxyl radicals by glutathione results in the formation of two water molecules. If reactive oxygen species levels exceed the ability to be broken down by glutathione and other antioxidant activity, oxidative stress will continue to cause toxic damage in the cells. So, by adding up the information, Inhalation of gold nanoparticles likely causes difficulty breathing and potential lung damage. However, research is minimal at best and is greatly needed in the field before therapeutic industry is advanced. Although gold nanoparticles may be a little bit toxic, or have the potential to be toxic, they are still reasonable for therapeutics under proper research. Recent research with T cells has been very promising in cancer treatment and the drug delivery system to tumors. A lot is possible through the engineering of the gold nanoparticles. Human T cells were activated by being cultured with gold nanoparticles which were coated with special antibodies. The gold nanoparticles were taken up and internalized into the T cells. Interestingly, no toxic effects were observed to the T cells. Functionality and viability was perfectly normal. The activated T cells were able to transport the tagged gold nanoparticles to tumor sites where the gold nanoparticle payload of drugs could be released. Injection was used in this method and in vivo testing has been produced on mice. The toxicity relative to the therapeutic potential of gold nanoparticles is still questionable. Based on studies, there is a lot of controversy and unknown information using gold nanoparticles. One thing is for certain, more research is definitely required.